高老师，谢谢你介绍的非常好。我今天也非常高兴可以在这边第一次做这个报告，所以I feel very honored. So thank you very much. As you know, my Chinese is not good enough for giving lectures. So I'm going to talk to you today about basis elements of intelligent primate systems. Let me see. Okay. So my purpose today is to give you some idea about what I've been Okay. So worked out idea, but I've collected some data over the years that have given me some thoughts about how the brain is so systematic, very organized, and that maybe we can understand an architecture of an intelligent system. Okay, so I'm very excited about this idea, but I don't know how long it will take to really make it into what I call a theory. Okay, so anyway, I'm going to talk a little bit today, but forgive me if it's not a fully fleshed out idea. Okay, so let me start. This picture is from our recent paper. The Hu Jiaming is the first author, and I will tell you more about this picture later in this lecture. So for any system, so you know I used to study math and philosophy, and we talked about systems. And so for any system, there are elements, there are axioms, there are theorems. In the brain, elements can be neurons or glia or vessels, and theorems can be rules about how these units operate. Hodgkin-Huxley, we know about Hebbian plasticity. Networks we know less about, but we still have some ideas. But I think we need to work in this direction to find out what are the rules for networked function. So I'm going to focus on this today. So let me just start by talking about what we know about brain architecture in primates. How is the information organized? And secondly, how are the networks organized? So primates have a large behavioral repertoire, and there's this idea in comparative science, comparative biology, that larger animals such as primates and humans have larger behavioral repertoires, and that's why they have larger brains, and so that kind of thinking exists. And we know that in order for a biological machine, such as the brain, to represent lots of information, the denser the information becomes, the more likely that one will need to make that information represented in a very organized fashion, and also in a very efficient fashion. So just give you an analogy, we have rural places in the world, and we have big cities in the world. And so if you live in rural places, you have much choice about where to put your house and where to put the roads, and there's not so much constraint. But in big cities where it's crowded, those constraints are very strong, and so things necessarily become a bit more organized. And so this is just a sort of depiction of, if this were a brain, the sphere here, that with low density information, you might not have as much systematic architecture, whereas with high density, the organization of information needs to be quite well organized. By the way, if you have any questions, please let me know. Just raise your hand. Okay, so if it is organized like this, and we have lots of information to represent, I've just chosen a few different parameter spaces here: color, motion, people, language, and trees. Just by random, I chose those. If you want to represent those parameter spaces in this on this surface, it likely has to be done in a very organized fashion, and you have to find space for your for yourself, if you are a tree, you need to like figure out which space you get, right? You're competing for all this space with other types of parameters. So that kind of competition idea is traditionally been linked to Hebbian hypothesis, right? Competition. And so this kind of idea has really dominated neuroscience and has led to some of the organizations that I will show you today. Now, the other thing that has to be organized is the networks. That is, the networks is what underlies behavior, not single nodes. It's entire networks, brain-wide networks. And so how these networks are organized, these many, many networks, is a challenge. I mean, how do I put all these networks into this one space? So that's something that is not so well understood, I would say, even mathematically. Okay, another idea I'm going to talk about today relates to sparseness. So sparse coding has been, this idea has been around for a long time. So for example, recently there was a report by Greg et al. In which they compared the sparsity of synapses in primates and mice. Okay, and so what they found is that primates seem to have somewhat sparser synaptic density on excitatory neurons than mice. And so they argued that this was something related to the need to be efficient, that you can't waste synapses. Every single one has to count. This is another paper talking about spiking sparseness. That is, in awake monkeys, this is an awake monkey study by Jack Gallant and his colleagues. In awake monkeys, what they discovered that surprisingly, the number of spikes fired to stimuli was very, very few. I mean, they were surprised because in anesthetized animals, neurons fired robustly to these stimuli, and it was with many spikes. But in awake animals, all of a sudden, they saw very few spikes. So what's going on? So they hypothesized that this kurtosis, as they call it, is something related to efficiency. When you are in a awake state doing a task, you need to be very controlled. The baseline is different. The whole brain is in a different state, and so therefore you don't want to waste spikes. Every spike has to be meaningful. Okay. The third paper I chose is this paper by Charles Connor, who he recorded from V4. Let's say he used Utah array and recorded 100 neurons, and he studied the response of every neuron to different kinds of curves, different orientations of curves, straight to very highly curved. And what he found was that for any shape, that the representation of that shape was primarily for the high curvature regions. That is, the places where the information content was highest. In other words, for a straight part of the shape, you don't need to represent that again and again. It's the same information. But it's the highly curved regions that really need a higher density of representation. And so they hypothesized that actually what is really represented is the salient features. And maybe the salient features is enough. You don't need to represent every single point on the shape. Just the salient parts. Again, another way to be efficient. So this idea of sparse coding has been around for a while. What I want to ask today is the organization of these columnar networks, as I will show you, is this kind of organization also sparsely represented? So I will come back to this idea throughout the talk. Okay, so let's just start first by how is the information organized in the brain. So we know from lots of many decades of work now that the cerebral cortex, starting in the visual system, has a very systematic organization for different features, including orientation, ocular dominance, and color in area V1. And this is something for which Hubel and Wiesel got the Nobel Prize. And then Pashko-Rikish showed that this kind of organization is predicted by developmental organization. That is, there are these proto-maps already early in development that become these very organized columnar maps in the adult. And so these ideas have been studied again and again and further extended and strengthened by other methods in neuroscience. So this idea is further further linked to our perception. So what does this organization have to do with behavior or with perception? And so Hubel and Wiesel and later Marge Livingstone showed that these specific units actually represent different aspects of what you see. So, for example, this red unit here is called a blob. It contains cells that respond to color information and represents surfaces with particular colors. These blue regions contain cells with different orientation preferences, and these are called orientation columns, and they represent border information. This is vertical, this is horizontal, and so forth. And then these two eyes, right and left eye representation, are segregated, especially in layer 4, and that information is used to calculate depth. So this surface is in front of the background, and the two eyes are needed to calculate that disparity information. And so this unit of surface, contour, and disparity information is what has been identified as a hypercolumn. This term has been around for a long time as well. The hypercolumn is a unit of cortex that represents, in their language, everything you need to know about a point in space. Okay, so we've accepted that, and that seems to be very strongly supported in area V1. We go on to other areas, and I'll be talking about that soon. So in V1, we can see all sorts of maps using intrinsic signal optical imaging. I've been studying these for many years, and we can see ocular dominance maps, we can see orientation maps that encode shape, color blobs here, as well as motion domains here happens to be V2. So early visual areas can be imaged in this fashion, and by interpolating this information, one can infer what an object might be. So with this kind of encoding, we can actually have a good sense of what is out there. So we have actually a paper, which I haven't put in here, but of a dot moving in space, and you can see this activation moving across the cortex. So the cortex very systematically represents what is out in the visual world. So this is just a summary table of what we know about organization of columnar organization in V1, V2, and V4. And so in V1, this is just a summary of what I showed you, the color blob, the orientation domains, and then what kind of features are represented by these units. In V2, we also know a lot about orientation maps, color hue maps now, higher order orientation maps, and then disparity maps, near to far disparity maps. And so the area V2 represents a lot of these higher order aspects of our vision. They are considered higher order than V1 because these cells can respond to many different cues that induce, for example, a vertical percept. And so then we get to V4, and in V4 we see that there are also organizations with these columnar units, and then organized into larger color bands and orientation, curvature and orientation bands. And so this is again a higher order area, and the units recorded here tend to encode color constancy, or curvature and other aspects of shape. So there is a very sort of systematic column-based way to view the organization in these areas. So let me just add now that we have shown recently that there is this kind of curvature representation, these maps from low curvature all the way to high curvature, that there are systematic maps in V4. And moreover, that these curvatures in different orientations are also systematically mapped. And so if you combine these two ideas, we can propose an organization which is called a curvature hypercolumn. So again, this kind of hypercolumn idea exists across multiple areas. In fact, there are similar principles of organization. So when I think about V1, V2, and V4, I think about these systematic maps, and to identify whether a feature is highly curved or has low curvature, you simply play a key on the piano. So this is what this picture is representing, where we have a piano keyboard here, and you play a key, and that's the curvature that you get at that location. Okay, so that's sort of a simple way to think about it. Okay, so let me just move on to one more question regarding columnar representation. So that is, in our vision, one very important aspect of our vision is the center of our vision, our foveal vision. And we recently looked at this region very closely. So this region of your vision is involved in many key aspects of your behavior, including visual attention. When something catches your attention, you move your eye, you move your fovea to look at that object. It has the highest density of photoreceptors, and the RGB cones are there, so you have color vision that is mediated by the fovea. Your fine manipulation, with your hands and fingers, that's guided by foveal vision. There are other concepts, and also in particular looking at faces and communicating with your eyes, whether you're angry or sad. This is also a very big part of your foveal vision. Okay, so what has been known about foveal representation has been done. Well, this is an fMRI study. There has also been electrophysiological studies, but this is an fMRI study. Let me point out that this foveola is the very center of the fovea, and it only represents 1 degree of visual space, which means if you stick your thumb out like this at arm's length, that thumb is occupying 1 degree of visual space. And so that amount of visual space is what your foveola is seeing, and that's what you use to update. So every time you move your eye, you're updating your picture of the world. And if you don't move your eye towards that, you don't update, and you have old information. Okay, so this is called the foveola, and it represents 1 degree. It's a teeny tiny part of the retina, only 0.3 millimeters of tissue. Teeny, teeny, tiny. Okay, in the cortex, how much space does the representation occupy? Well, there are studies here that show that this is the foveal center, and that they call this a foveal confluence, where different areas, the fovea representation of different areas come together. This has led to a lot of controversy. So some people believe that there are separate foveas for different cortical areas. Some believe there's this one giant region that represents fovea for every area. And then some believe that even some cortical regions, such as V3, do not even have a fovea. So these are some controversies that have not been resolved. So we decided to look at this question in monkeys, which are trained to perform fixation, very excellent fixation in the 7T MRI. And so what we found, so these monkeys were trained for a long time. Also, some of the technology that was developed specifically for this, the RF coils, 16-channel RF coils developed by Zhang Xiaotong and Gao Yang, were absolutely necessary for this project to even begin. And then Wang Jianbao also developed many data analysis and surface view methodologies that enabled us to see this. This kind of organization is not necessarily easy to see in slice view. That means slice by slice, you might not realize there's this representation there. But what we found was that using tiny spots that the monkey was fixating on, we would get activation, and these are different thresholds of significance. So the highest threshold of significance is the blue color, and you can see that in V1, V2 region there are a couple locations of blue, V2, V3, blue here, and we have done many studies. This is highly reproducible and very reliable in both of the monkeys that we looked at, and now we are looking at a third monkey. And so for each area we found that there were foveolar, this is foveolar, so teeny tiny stimuli, foveolar activations, and that they formed a ring of foveolar representations. This is something that has never been seen before, and it's not been seen because there hasn't been the technology to see it. So now with the 7T and ultra high field spatial resolution, this is actually 0.6 millimeters in plane, so quite nice resolution. And then with the coils that boosted SNR, we were able to see these activations. And so what this tells us is that there is a region here. For every cortical area, it has its own foveolar representation. And in fact, the dorsal and the ventral portions of those visual cortical regions have their own foveolar representation. But in addition, there's this cortex in the center here. And this is a mystery right now. But this center area is huge. It's huge. And it absolutely is the center of our vision. So it's the very lateral part of the brain, and all these foveolar zones are likely to integrate together to calculate some higher order aspect of foveolar vision. Okay, so that's something that we continue to study. We've gotten some very nice values for foveolar cortical magnification that hasn't been known before. And so this is something we're quite excited about and hope to further pursue. Now, I had asked the question whether foveolar cortex also contains columnar organization. And so here's some preliminary data that indicates that the answer is yes. So here we see V2, and you can see these V2 stripes here. These are color stripes seen very regularly as expected in V2. Down here is the foveolar region, and then you can see anteriorly in area V4, you see beautiful color patches. These are color patches, and this is enlarged on the right here. And you can see the red patches are color patches, and the blue patches are luminance, dark to light patches. And so there's a very nice organization of columnar representation, and you can see some of these are in the foveolar region. Secondly, so this is part of what's called the ventral pathway, the pathway for object recognition. Now, the dorsal pathway is processing vision to enable you to navigate the world. That means to open a door by grabbing the doorknob, to pick up a mug and shape your hand in the appropriate way that your vision instructs. And so this dorsal pathway is very important for self-motion, navigation, and movement of your body parts. And so here we find that area MT, which is difficult to study in many cases because it's buried deep in the sulcus, this also has columnar organization. And you can see here that each of these colors represents a different direction. And it's not been known what the organization is in a two-dimensional space. And also MST, we see some maps here in MST as well. So this is a single monkey. We have a single monkey looking at different visual stimuli, and we can see sort of global views of where these patches are and what kind of functions they have. And so this is something that, again, is the first time at this spatial resolution being able to see these organizations. Okay. So let me go on to talk about what I think the encoding of objects might be. So here I have used the term columnar code. Now the term column is very, it has a different meaning for different people. In V1, the classic column is a vertical 6-layered column where the cells within the column is typically for orientation, and so that is the classical definition of a column. Now, in other areas, maybe the word column is not quite appropriate, but they're units of functionality where cells share similar functionality. The other thing I would like to point out is that often people think, oh, a column is this anatomical structure, and they're like bounded units. That is not necessarily true. So in V1, orientation columns shift continuously as you go across the cortex. So it's not like there's a boundary between, you know, a 10-degree orientation and a 20-degree orientation. It's sort of shifting in overall preference. And so one can think of these columns as discrete entities. Here I've drawn them as discrete entities, but they can also be thought of as continuous variables. Okay, I just want to make a question you want to answer. One representation may be a little easier than the other. So the basic idea is that every cortical area maps variables, right? We've talked about color, we've talked about disparity, we've talked about orientation, curvature, and so each area has a small handful of variables that's really keyed in on. So every area is looking at the same object, but paying attention or encoding specific aspects. So every area is coding a different aspect of that object. Okay, and so in V1, the idea is that V1 is really good at spatial location. It has a really fine resolution for where things are in XY coordinates. And so V1 provides the best information on location. V2 has maps I've drawn two here for disparity, near to far disparity, and for hues, blue to green, yellow, orange, and red. So there are hue maps in V2 in the thin stripes, and there are disparity maps in the thick stripes. In V4, there are other variables such as orientation, curvature orientation. So these are other variables. I mean, there are more variables than these. I've only chosen a couple to show. But in my experience, these variables are probably 3 to 6 in number. At least that's what I've seen so far. I'm not saying that more can't be found, but I also think that there is a limit as to how many variables can be systematically and in an orderly fashion represented, and in a complete fashion represented in a single two-dimensional cortical area. Each column is one value. So as I said, you're playing the keys on the piano. So if you're playing this column, you get that value. So if you wanted to, let's say, have a representation of this corner of the triangle here, okay, you would play this key, and the triangle is red. Then you play this key, and the triangle is in front of the background, and maybe you play this key, and then it's highly curved here, so you play this key, and the curve is oriented in this fashion, and so you play this key. So you have a set of keys that you're playing that represent this object. Okay, so this is what I call the object code. It's a columnar object code, and the values that are selected are simply a set of these values across cortical areas. So you may choose a couple or a few from each cortical area, and the entire set across all cortical areas is the code for that object. And if one has this and understands it, then this code would be so easy to decode. You don't have to do much computation. You just play the piano. So this is something that I think, at least at this level, a mesoscale level, is a code that is very accessible and also renders itself accessible to some mathematical hypotheses. Within these nodes, I do fully want to say that these nodes contain tens of thousands of neurons, right? Very dynamic, and but they share a job. They share a job. So that is, and I also think that people who record from single neurons, they're studying neuronal response, which is key, which is fundamental, but it's hard to see this kind of organization with that kind of approach. Okay, one thing that this idea, this layout here says is that the coding is really sparse. You don't need a whole lot of nodes, maybe 100 nodes, and that's it, 50 nodes, and you have your object. Okay, so this suggests that there is a sparse coding. There can be a very sparse coding. So I will come back to that later. So right now, let me just summarize this first part. We've shown that ultra-high field 7T MRI gives you enough spatial resolution to see these, exciting new structures in the brain. We can train awake monkeys to sit in there and have their brain scanned while they're sitting, and we are now continuing to train them on different tasks. The information from these maps we see are really organized, very systematically organized at mesoscale. We have this idea about what the object code might be. And then one new finding is that the foveolar representation is just enormous. So for a 0.3 millimeter piece of tissue in your eye, it occupies like, 1.5 to 2 centimeters of the cortex. It's huge. It's very substantial. So that cortical magnification says that our foveola is very important to our survival. It's involved in many important functions. Okay, and I also think that because foveolas are only seen in primates and in humans and in some primates, that this is a specialization of primates. Okay, this is a special area of the brain, just like a bird has a representation of a wing, and bats have echolocation. Those are specializations. So I think the foveolar representation is a specialization of the primate cortex. Okay. All right. So I drew these circles here just to show that V1, V2, V3, V4, etc. have different size spatial scales of receptive fields. And so they're all coming together here to calculate something that crosses spatial scales. So this is a question that I hope somebody answers. It's a very intriguing question. All right. Any questions so far? Yes, Jingwei.
Thank you. Very beautiful data. So I'm interested to know when we see the activation of different columns, and does that mean the function was involved into multiple columns, or was that so how much was this connected with the relatively rigid anatomical traditional oculomer structure?
Yes, yes.
So this is point one, question one. And question two is, in terms of visual cortex, and how much difference between the other brain region, like motor cortex, primary motor cortex, between that and visual cortex? Thank you.
Okay. Yes, these are excellent questions about anatomy. There is a very specific anatomy in the brain that is also very systematic, and we are learning more and more about this because, as I will talk about soon, we have a method for evaluating these connectivity patterns, and they appear to be extremely systematic, and much of it matches what we already know about anatomy, although the known anatomy at this mesoscale is not so much. I mean, the available data is not so much. But the data that is available says that yes, what we are studying is very, very similar. We're revealing similar circuitries. And so that's the answer to the first question. The second question is, and I want to add that anatomy is not, that doesn't limit the dynamics of the system. I mean, there are still can be much dynamics going on. Okay, if you have a ball with multiple nodes, you can turn off one node and you can activate another node. So there's a lot of dynamics that can still be achieved. The answer to your second question is it ubiquitous? Well, if you believe Pashko-Rakesh, you would think so, yes. In my experience, every place that I have looked in the brain has had these nodal organizations. And if it hasn't been seen yet, I believe that it's because we haven't presented it with the right stimulus. But that organization is still there. So in motor cortex, as you say, it may be harder to see because we don't really understand what is the basis for that organization. Is it a muscle unit, or is it some joint receptor? So there's a lot of unknowns there. But there is now emerging some evidence from fMRI that there are functional domains for this kind of movement versus this kind of movement. Okay, so there are sort of growing pieces of evidence that motor cortex may also have this kind of organization. Okay, thank you for that question. Excellent. Okay, so let me go on now to talking about networks and their organization. So one of the projects that Hu Jiaming was involved in, or he pushed this entire project, is using microstimulation to look at local connections in the cortex. And in particular, he stimulated different nodes within V2. So he did optical imaging first of V2, and then determined where are the thin stripes, the thick stripes, and so on and so forth. And here is a summary of one of his cases where these red dotted lines are outlining the thin stripe activations, and these white and orange regions are domains. So these are color domains within the thin stripes. So he conducted color minus non-color imaging, also compared with orientation maps, and the orientation maps fell in the inter thin stripe regions. So what he did was stimulate one of these domains in the thin stripe, and he saw these activations, which were confined only to thin stripes. Okay, very, very specific. And when he did the same thing and he stimulated in a thick stripe region in an orientation domain, he saw activations in other nearby thick stripes that not only were in the thick stripes, but furthermore, they matched in orientation selectivity. So this has never been seen before by any method. So this is something that is says very fundamentally that the functional specificity is really high. It's these networks are very functionally specific. So you might ask, well, why are they that way, or how did they get to be that way? And that may be a developmental question. I'm not sure, but the fact is they are highly segregated. These stripes are highly segregated, first of all, and then their connections are highly specific. All right. So another aspect of this connectivity that he discovered was that if you move the stimulus from one location to an adjacent location, this whole lateral network shifts with you. Okay? You shift this point and you get these activated. You shift this a little bit here and you get these activated. So shifting the stimulus point predicts the location of the activated site in the connected regions. Okay? So that's quite remarkable. So that means networks are topographically organized. They're connections are topographically organized, not just the nodes. Right? Okay, this is something that would be difficult to study with just anatomy, because we can look at multiple sites with this kind of stimulation approach. And then finally, we observed that activations in V1 here were also quite similar in layout. So if you stimulate a orientation domain in a thick stripe, you see connections to the activations in these intrablob regions, the orientation domain. If you stimulate a thin stripe, then you see these activation in blobs, the color domains. And the structure, the architecture of these connections are very, very similar. The convergence, divergence seems to be similar. So the number of units that you activate seem to be similar. So what we see is that from a single site of stimulation, you get lateral connections activated, you get interareal activations activated. And this kind of lateral and interareal architecture is shared by color and orientation parameters. All right. So this is what this picture represents, the first that was on the first slide. This represents a little micro network here, okay, connecting like stripes in V2 and connecting appropriate columns in V1. Color and orientation share the same network architecture, okay? So these are canonical networks that are shared across functional stripes. They're shared across different topographical locations in V2. And I believe that we could say they comprise cortical circuit elements. That means of the whole brain network, this is a little piece. This is a little piece that is repeated, right? It's a fundamental, like, basis vector. And you can create larger circuits from these little pieces. You know, it's like when you were young and you had these chemistry sets and you were making molecules, and, you know, you can make lots of things from these little pieces. So maybe that is something that we discovered that is, you know, the beginning of a crystalline structure, so to say. All right, so let's move on here. I have showed you that in this local region, there seems to be some repeating network pieces. Now, what about a whole brain scale? Whole brain scale is much more complicated. So for this question, we developed a new method of stimulating single nodes in the brain and then mapping brain-wide networks, mapping node activation nodes at brain-wide scale. And this was done in the 7T MRI using infrared neural stimulation. I'll tell you more about INS in the next slide. But for now, I just want to make a point that this method is very powerful because you don't have to sacrifice the animal. This is done in vivo. The animal is sitting in the MRI. It's done in vivo. It's fast. You see the activation right away on your screen when you stimulate. It's at mesoscale, so you can stimulate sub-millimeter nodes and see mesoscale activations elsewhere in the brain. In addition, there's no reconstruction. You don't have to align sections. You don't have to, you know, do. You simply see the brain right there, okay, on the screen, and you see these nodal activations. So there's no reconstruction involved. It's quite direct. You can study multiple networks in a single animal and compare those different networks. And finally, we are now studying change in single networks over time in single individual animals by stimulating, repeatedly at different sessions. Okay, so let me just give you an idea of what INS is. So here we are looking at the effects of INS stimulation at the cellular level, at mesoscale optical imaging level, and at whole brain bold fMRI level. So here is some calcium imaging. The INS fiber is placed here, optical fiber, and the parameters that we stimulate with are, in this study, there was 6 pulse trains. All right, so each pulse train is half a second, and each pulse is only 1/4 of a millisecond wide. So they're really very brief, beep, 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 like that, okay, very brief, and it's pulsed at 200 hertz for half a second. And so one of these represents a half a second pulse train. And he presented 6 of these pulse trains. And each of these pink lines here represents one of these pulse trains. So you can see each pulse train induces a very, very clear modulation of the calcium signal. This calcium signal is going up and down with each pulse train. And then, so this is clearly modulating neuronal activity. And moreover, they labeled GABAergic and non-GABAergic neurons. So they could identify this activity in both types of neurons. Both GABAergic and non-GABAergic responded in this fashion. So we're confident that the INS is influencing the neurons directly. This is hard to ascertain with electrophysiological methods because the light can induce electrical current in the electrode directly. So there was always that ambiguity. Well, is the current due to the light or is it due to actual neuronal response? So this approach, I feel a little more confident that neurons are actually being directly activated or modulated by the light. With optical imaging, and also this shows that there's an intensity dependence. I didn't put the graph here, but as you increase the intensity, you get a larger amplitude of response. With optical imaging, again, we see this very clearly from 0.1 to 1/1 joule per centimeter squared of energy. You get a larger and larger optical imaging response. With bold MRI, also as well, stimulating V1 here, you can see that as the intensity increases, the bold response increases, and that's what's graphed here. Okay, so all of these show that the INS is having an effect on the neurons and that it is intensity dependent. So one of the tests that we used to ascertain whether a voxel in the brain was connected to the stimulation site is to see if that connected site is intensity dependent in the same way. So that is a very useful test. So I'm just going to mention this very, very briefly because I've talked about this before in previous talks. We have done a connectome of amygdala stimulation sites using this stimulation method in the MRI, and we've shown that single sites in the amygdala can activate patches or columns of activation in different cortical regions as well as in subcortical regions. So we see that these networks are very, very specific again. So these patches match the motor maps, the limbic motor maps known to be in area 23 and area 24 in cingulate cortex. So it's very specific, very sparse. Again, you see that there's not a lot of voxels here, and they're very specific. They're mesoscale in nature. They also shift as you shift the fiber and stimulate it continuously at different sites. You get shifting of this whole global network. So that's remarkable. Again, okay, we see the same thing we saw at the local level, and then finally the total nodes that we see from single site stimulation suggests that these are behavioral networks. So, for example, we see nodes in brainstem and hypothalamus, and when CE was stimulated, that suggests that this has to do with food acquisition or other types of social behavior or navigation. So, the locations of these nodes that are activated really tell a story about what the circuit's doing, and is at brain-wide scale. Includes limbic, motor, sensory, and physiological function locations. So, anyway, it's very exciting to look at. Okay, so let me tell you just very quickly about our more recent project, stimulating in the pulvinar. To answer the question, pulvinar has been a very mysterious place in the brain. It's thought to be involved in attention and salience, but where there are 3 primary subnuclei in the pulvinar, PM, PL, and PI, and it's not very well known what the internal organization is. There's Toru Takahata has published a paper recently showing that there are more maps than previously thought in the pulvinar, and so there are many unknown things about pulvinar. So here again, we delivered 4 pulse trains, and you can see that every pulse train led to a very nice bold modulation here. And this is intensity dependent. Increase from 0.1 to 0.3 gives you a larger and larger bold signal. So these are the total sites that were stimulated in 2 animals, in the blue animal and the red animal, a total of over 30 sites. And so I'm just going to show you one slide giving you a sort of a taste of what the whole brain activation looks like. So you see these voxels here. They're all the way from the front of the prefrontal cortex all the way to back of visual cortex. And you can see that these are patchy activations, very, very specific. If you look at these closely, you will see that some of them are highly topographic, and you will also start to understand what the circuit is involved in. Okay, and we can label these areas. We can know what areas they are according to monkey maps that exist. All right, so here is just one example of topography of PL and PM. And so you see we had one penetration here that goes down. This number 11 is in PL and 12 and 13 are in PI. And you can see that 11 here, 12 here, and 13 here. As the fiber stimulation site goes down, you see the activation shifts from peripheral V1 to around 10 degrees in V1 to foveal locations in V1. And furthermore, the V1 activation here has reflection across the border to V2, and V2 has a reflection across the border to V3. So these are just gorgeous topographically appropriate activations from single stimulation sites. So, and furthermore, these colors are different p-values, different thresholds, and you can see that across multiple thresholds, the center of the locus stays stable. So this shows that this is a reliable and real site. It's not a artifactual activation. So this also gives me a lot of confidence in this method, and Toro and Yao Songping have done a lot of work to put this onto flat maps so that we can understand the organization more easily. Okay, and again, these circuits are really specific and really sparse. Okay, and they also have compared connectivity of PI, PM, and PL to dorsal and ventral pathways and found some very nice results as well, but I'm not going to have time to talk about that today. Okay, so let's go back to sparseness. We were talking about sparseness a while ago. This is all the slices from, very caudal to well, not very anterior, but all the visual cortical areas are in here. Okay, and what was stimulated is a single point in B1. Now, what do you see? What you see is very, very few voxels, and every voxel is in a different area. What this suggests is this one stimulation point activated all these visual cortical areas, literally all the visual cortical areas in both ventral and dorsal pathways, but highly, highly specifically. Just one voxel, right? And so for most MRI people, they would do, Bonferroni correction, and then you lose all this data. So you cannot do Bonferroni correction with this kind of data because single voxels will just, disappear. So we have done other types of permutation tests and other types of statistics to convince ourselves that this is really true data. And we've also, so that's been published in Neuroimage. But more compelling, I think even more compelling, is the specificity of these activations. So what this says that this one node here in V1 is connected to all these visual cortical areas, and there's a network that integrates across areas, a nodal network that integrates across networks, and that you can activate this whole network with one node of stimulation. Is that remarkable? So if I touch this one node, I can maybe modulate the whole circuit across the brain. If I take away that node, maybe I will change, what all those other nodes are doing. So this is a basis for a dynamic system. Okay. I'm getting too excited. Okay, so yes, go ahead. Uh-huh.
In order to increase the contrast, actually, all the information was taken, but only one stand out after calculation.
Uh-huh. I see. Okay.
What does that mean?
Right.
First, everywhere almost was activated, and then if the visual is actually very accurate, what does that mean?
Okay. Right. Excellent question. So let me remind everybody what I'm doing now, just stimulating one point with INS. It's not normal. I mean, that's not what we experience when we look in the world, okay? When we look in the world, the whole retina gets activated. But what does happen, I believe, is that our brain narrows down to the most important aspects, the most important points, okay? The most important columns for that particular moment, for that particular behavior. And that what this is showing you is that when you zero down to those small set of points, it's accessing actually relatively few points in the brain to achieve what you need to achieve for that specific behavior. So I don't want to give you the impression that, you're only activating one node at a time. That's not what I believe. But what this represents is that if you activate that node, you are affecting a small set of nodes across the brain in a very selective fashion. Okay? Now, in a full picture where, your retina is being stimulated by people and cars and everything, it's going to be a different situation, but I think our visual system is efficient, and so therefore ignores a lot of that. And that's why you have sparse spikes, right? And that's why you have sparse population coding, because you don't want to waste all your energy encoding less relevant or less important information. With this INS method, you mean? Good question. I cannot give you an answer to that. It's a very good question. Because what we see, we stimulate here, and these neurons are activated, and they send their axon to another place, and those neurons are activated. So by definition, that has to be an excitation. Okay? Whether that also leads to some local inhibition in the target zone or even in the stimulation zone, I believe there is, because if we increase the stimulation up to a certain point, beyond that point, we will start to see the suppression of the signal. So we're probably then engaging more inhibitory, the excitatory inhibitory balance has changed, and we're engaging more inhibitory circuits. And so, but that's kind of hand wavy. We would have to study that very carefully. But let me say one more thing. We know that if you stimulate a single orientation domain in V1 and do imaging, let's say the orientation is vertical, you will activate or modulate, enhance the activity of the vertical domains nearby, okay? And you will relatively suppress the activity of orthogonal domains nearby. So it's like a push-pull situation, right? Stimulating one site has both excitatory and inhibitory effects, but on different domains. And at a brain-wide scale, I think that's probably also the case. You know, there's a lot of examples one could bring up for that. But that's something we want to really investigate. It's so important, that question. And we need to do that more in a behaving animal. Yeah. Yeah. I'm sorry. It's hard to hear.
Kind of connection, are we born with this kind of connection, or are these trainable connections between different places?
Generally, I would answer it with a yes, but that doesn't mean there's no plasticity. I'm sure there's plasticity involved, and depends on what kind of plasticity you want to consider. During development, there can be a lot of plasticity, right? Synapses can be removed and connections lost and all sorts of things. In the adult, I believe that it could be the synaptic strength could be modulated, potentially even synapses could be lost. You know, there certainly is plasticity in the adult as well. Is that your question?
Yeah.
Yeah. So, is it hardwired? I think your question is everything hardwired. To some extent, yes, the wires are there, okay? But how you use the wires can be still very dynamic and allow you to resculpt the circuit. You know, I believe a lot of that happens. But I guess the anatomy does constrain what the total possibilities are. I don't believe that you can access every point on the brain from one point. I believe there is some specificity there. Okay. I am going on too long. Okay, let me kind of quickly finish this. This is just some computation. My colleague Yu Guo, who is at Fudan University, he did some calculations. So he took this data. Oh dear, I had a very important slide here. Okay. Let me see. Is that not? Okay. Okay, well, I'll have to show it later. So here we see some calculations where you have a non-similarity index. That is, if you compare the network of nodes that is activated by stimulation from one site with that in another site, how similar are those two networks? And so if they are very similar, then they will be overlapping. You have a lot of redundancy. If they're not similar, then they're encoding something, each encoding something very different. And so you do this kind of pairwise statistics and you come out with a non-similarity index. And an index of 0 means there's a high redundancy. An index of 1 means there's very little redundancy, and that it's exceptionally sparse and, well, very efficient, okay? So you're not encoding the same information again. That means these networks are non-overlapping, largely non-overlapping, okay? So of the 200 points, I had another slide that was in here. Of the 200 points that we stimulated in the visual system, he did this calculation of, what sets of nodes were activated and calculated their similarity index, and found that indeed the voxel stimulase encoding is very, very specific and very, very efficient. And this is another calculation based on not voxels, but based on areas. And he found again that the similarity is non-similarity index is very high. And so this means that the stimulus coding is very, very specific and non-redundant. Okay? And this is not true in lower animals such as aplysia or zebrafish. That I haven't looked at that myself, but that apparently is true. Okay, so we're working more in this direction to try to understand sort of more global, quantify more global aspects of the networks. Okay, so let me just finish this. Maybe I won't talk too much because this is already over 1 hour. So, right? So let me just go to the end here. So here I have this idea of axioms and theorems in the brain, and this I think is the beginning of a new thing. Okay. So this is a representation of the brain and the connections between the brains. Each brain area has a set of nodes, and the connections between two areas are basically 2D to 2D mapping, and so it's a matrix. And so what we're seeing here is we have a node code, what I call the node code. Okay, x1, x2, x3, so far. Selection of values from different parameter maps. The elements are these micro networks, and that they might have different types of motifs. And then thirdly, local and global circuits, somehow they optimize so that they have efficient wiring, so you don't waste wires. So what is that calculation that gives you efficient wiring? That remains a question. And then finally, the dynamics. So we have known Hebbian dynamics for quite a while. You know, the idea that if you're competing and you have two synapses coming in, if they co-activate a postsynaptic neuron, then that strengthens the synapse, whereas if they are, firing randomly, then the synapse will weaken and those connections will go away. So this same concept can be applied. So that has established our understanding of how we get ocular dominance columns in V1, how you get orientation maps in V1, but we haven't applied it to how connections are established. Why not? That needs to be done. There may be one group in Germany who is trying something like this, but I feel that this is a big question that needs to be addressed. And people who with mathematical and computational background, probably not so difficult for them. So I'm hoping somebody will tackle this problem. Okay, so just summarizing, we've seen that in the primate brain, brain-wide networks are based on mesoscale nodes. We see this at both local and global scale. Networks are sparse, they're efficient and non-redundant. Remember that non-similarity index. And then micronetworks may comprise fundamental elements of this larger global network. And finally, I hope that we work towards a mathematical representation of circuit elements. Okay, let's skip that. And I want to thank a lot of people. This slide is not big enough to fill all the people that I should say thank you to, but people in my lab, definitely who work like tirelessly for all days and nights. I have to thank them, thank the other PIs in the Institute who have, really made our Institute interdisciplinary. Great thanks for the people who are engineers, working with people who are biologists, working with people who are optical engineers, lots of collaboration. And of course, I thank all the important people at Zhejiang University who have helped enabled this project to come to life. And I thank funding agencies and everyone in my lab. Okay, and I thank all of you for listening to this long talk. And thank you for your questions. Okay, please feel free to ask questions now. Let's see.
You mentioned the connection in the primates. I was just wondering, is it really unique to the primates that the connections are sparse, or does that also occur in other animals?
Yes, so...
And how sparse are they compared to human
Right.
and mouse? Depends on the density, like what's the ratio?
Right, that's a great question, and I don't have the answer. I don't think there is much data on this. So there's that paper I pointed to is one of the few papers that really have directly compared the same thing in mice and in monkeys. So we don't have a good sense yet of what is the degree of sparseness, but I can say at least that we know that in mice there are no cortical columns. There are no even in cats don't have color, for example. So their domains are, in some sense, less constrained. They don't have to put as many things in the same space, and so their domain architecture is just a little different. It's not so systematic, not quite so systematic as in monkeys. And then I would love to see in animals, in humans, in whales, whether they have even greater constraints,
Yeah.
So it's a great question.
I was also wondering, that this kind of sparse connections, of course, it's kind of like representation of the number of synapses, right? Or, I mean, is it comparable?
Well, so, according to the few papers I've seen, there are differences between primates and rodents.
Okay.
And the order of magnitude may be, 30-50% kind of order of magnitude. It's not huge, but still, it's there.
Yeah.
We may need to look at this much more closely. In terms of nodes, though, I think that can be supported. We do have data from different types of monkeys, from cats, and we also have some rodent data available. And so I think that kind of data does support this idea.
Thank you. I was also wondering, because you tested this visual system. Of course, those monkeys are very alert with their vision. But on the other hand, they are very active in climbing, right? Climbing up the trees and also suspending their tails. So I was just wondering, what about in other cortical region, particularly for those that are in charge of this motor activities? Do they also have this similar connection pattern?
Yeah, very good question. We don't really know. I think there are only a few MRI studies that suggest in human motor cortex there might be these columnar organizations. In somatosensory cortex, definitely we see these kinds of organizations. So, yeah, excellent question. That will take some more research. Probably electrophysiologists don't necessarily see this kind of organization in motor cortex. But, this is a population level organization.
Yeah.
Right.
So, Anna, I have a question that for, monkeys and humans, the brain is still slightly different in some regions. For example, vision system and the other very important decision-making systems. How do you think for human beings, how is there a better way? Because INS for humans, it's still a little bit difficult to test. Do you have any suggestions on how to measure the human functions in the future?
Well, I would encourage pushing the ultra-high field imaging, because if we could get to columnar level imaging in humans at brain-wide scale, then we could study, resting state at this mesoscale level, and that might be a really powerful way to access some of these important questions. Yeah. But yeah, you're right. We can't easily stick probes into people's brains.
Thank you.
Thank you for an excellent talk. And I was wondering that you mentioned that when you stimulate a vox in the visual one cortex, and you have found that in other visual cortex there also one vox excitation point. So it means that there is a delicate connections between these visual cortex points, and this kind of connections is somehow very vulnerable. You know that if you broke one point, the other point will be break also. So that if our brain is organized in this way, I think that our brain may, if it is a break in one point, the other point will be influenced. So that what would you think if it is organized in this way? What's the meaning behind these organizations, and why?
Excellent question. That's a very, very good question. So I will say something now that I didn't say because I didn't want to complicate things. But we saw that one voxel because of the threshold we chose, right? If we choose a lower threshold, we will see more voxels. And in fact, in the pulvinar image that I showed you, we showed different p values, right? And so at the lower p value, you saw slightly bigger activation, and at higher p values, you saw more focal activation. And so what I think is that, and I actually have written this in the paper, is that your baseline shifts with your purpose, or with your attention, or with your task at hand. And so if your baseline becomes more synchronized or more tight, then you may one voxel may already be enough. Okay, a tiny signal may be enough. But if you have this big oscillating baseline, then that voxel won't even register. So I think the other aspect of this encoding is to get to this level of specificity, the brain has to be in a certain state. And that is probably dynamically modulated. And I don't have any data that is relevant to that aspect.
So it may be that if one point is excited, so that the brain can be easier to accept this information and to excite other brain regions, so that it is excitation threshold may be higher.
Right.
Yes.
Right.
Yes.
So when you're really paying attention to something, or when you're vigilant about something, any tiny stimulus will,
yes,
you're going to pay attention to it, right?
Yeah.
But if you're kind of like sitting in the lecture hall and you're kind of falling asleep, even if I speak very loudly, you may not pay attention. So it depends on the animal's state. I think that is another big factor that we need to understand better.
Yes.
Thank you. Yeah, thank you for that question.
So if there's no more question, we Thank you very much, Anna, for this wonderful talk. And thank you, everyone, for attending this lecture series. We'll see you all together next time. And enjoy the summer. Thank you.
Thank you all very much. If anybody is interested, I'm going to get that missing slide and show you.
Where is it now?
It's on my computer.